Good evening to everyone. Myself Atharo Sandeep Firke. I am presenting presentation on DNA, RNA and its replication. So I am going to start now. So the first question appears: what is DNA and RNA? So we see here that DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid only. DNA is a molecule that contains the and instructions that an organism need to develop, live and reproduce. RNA is a polymeric molecule essential in various biological roles in coding, decoding, regulation and expression of genes. DNA is a molecule that is called the double helix. In molecular biology, the double helix refers to the structure formed by the double-stranded molecules of nucleic acids, such as DNA. DNA is one example of double helix. Um, so let's start the other. We have finished here with the DNA RNA. So we will start the topic about replication. Just before, who invented DNA and RNA? DNA and RNA was invented by James Watson. The double helix, the topic, which means the term, was given by James Watson. Personal account of discovery of the structure of DNA by James Watson, we say. So we will move on to DNA RNA replication. First, we will study about DNA replication. DNA replication is a biological process of producing of the two identical replicas of DNA from one original DNA molecules. It means reproducing, like in other organisms, like the cell division and other. Like in DNA, just the double-stranded, the two strands separate from each other to form two different DNA molecules afterwards when they grow. So DNA is made up of double helix of two complementary strands. During replication, these strands are separated. Each strand of the original DNA molecule that then serves as a template for the production of its counterparts, a process referred to as semi-conservative replication, which means the one strand will then further grow into one DNA molecule. So as a result, the semi-conservative replication, the new helix will be composed of an original DNA strand as well as a synthesized strand, which means the new formed strand, which is going to be the double helix again. The cellular proofreading error checking mechanism ensure near perfect fidelity for DNA replication. This is actually done in the artificial DNA replication. That the that cellular proofreading and error checking are done when the DNA is replicated. So in DNA replication, in a cell, the DNA replication begins at a specific location or region of the replication in the genome. So the question appears, what is genome? Um, in the field of molecular biology of genetics, a genome is a genetic material which is formed in the DNA. It consists of the DNA or RNA in a RNA virus. Actually, virus is consisted of proteins that are wrapped in a coat. So the genome includes both the genes and the non-coding DNA as well as mitochondrial DNA and chloroplast DNA. There are several types of DNA which we will study afterwards when we go deep into it. So here comes the structure of DNA replication. Here you can see that the DNA strands, which are a double helix which contains codes, are separating from each other and then joining a new DNA. These are arrows, but afterwards they join a new DNA strand and which they synthesize by themselves. So this is now for a